Good morning, students. We are discussing about the biomolecules. Today we will start protein. The last class we have discussed about the carbohydrates, types of carbohydrates, then lipids, there are different types of lipids, simple lipids, complex lipids, derived lipids. Now, third important biomolecule is protein. Protein is the compound of carbon, hydrogen, oxygen and nitrogen mainly. Proteins are the building block of all livings, and that is nothing but that is a chain of amino acids. So in today's topic we will discuss about the various functions of the protein and the structure. Proteins, most abundant organic molecules of the living system, they form about 50% of the dry weight of the cell. They are most important for the architecture and functioning of the cell. Proteins are polymers of amino acids. Proteins on complete hydrolysis yields amino acids. There are 20 standard amino acids which are repeatedly found in the structure of proteins, animals, plant or microbes. Collagen is the most abundant animal protein and Rubisco is the most abundant plant protein. Protein synthesis is controlled by DNA. Amino acids. Amino acids are a group of organic compounds having two functional groups, amino and acid group. Amino group is basic, whereas acidic group is acidic. R can be hydrogen in glycine, CH3 in alanine, hydroxymethyl in serine. In other, it can be hydrocarbon chain or a cyclic group. All amino acids contain carbon, hydrogen, oxygen and nitrogen, but some of them additionally contains sulfur. Physical and chemical properties of amino acids are due to amino, carboxyl and R functional groups. Amino acids are differentiated into 7 groups. See, the structure of glycine and tryptophan. These are the example of amino acids. Neutral. Amino acids with one amino and one carboxyl group. Example, glycine, alanine, valine, leucine, isoleucine. Acidic. When more carboxyl group is there in comparison to the amino group, that is acidic in nature. Aspartic acid, asparagine, glutamic acid and glutamine. Basic, when extra amino group is there, that is arginine, lysine. S containing amino acids have sulfur, cysteine and methionine. Alcoholic amino acids having OH group, serine, threonine, tyrosine. Aromatic amino acids having cyclic structure, phenylalanine, tryptophan. Heterocyclic amino acids having N in ring structure, histidine and proline. So in that way there are 7 types of amino acids. See the structure of various amino acids: alanine, neutral, aspartic acid, acidic, lysine, basic, serine, alcoholic, cysteine, sulfur, and phenylalanine, aromatic. Peptide bond formation. When two amino acids approaches that joins by losing one molecule of water and forms dipeptide and peptide bond is formed. So in that way, number of amino acids join to form a chain, and that chain is protein, chain of amino acids. Peptide formation. Amino acids are linked serially by peptide bonds formed between the amino group of one amino acids and the acid group of adjacent amino acid. Chain having two amino acids linked by a peptide bond is called dipeptide. The sequence of amino acids present in a polypeptide is specific for a particular protein. Means the type of protein depends upon the number and sequence of the amino acids. Structure of protein. Four basic structure levels are assigned to protein: primary, secondary, tertiary and quaternary structure. Primary structure. The primary structure refers to the number of linear sequence of amino acids in the polypeptide chain and the location of the disulfide bridge. The primary structure is responsible for the function of the protein. The N-terminal amino acid is written on the left side, whereas the C-terminal amino acids is written on the right side. Secondary protein. The folding of the linear chain into a specific coiled structure is called the secondary protein. There are three types: alpha helix, beta pleated sheet and collagen helix. First, alpha helix. Second is the beta pleated sheet and collagen helix. These are the example of secondary protein. Tertiary protein. The helical polypeptide may fold upon itself and assume a complex but specific form, spherical, rod-like or something in between. These geometrical shapes are known as tertiary structure. Quaternary structure. Proteins are said to be quaternary in structure if they have two or more polypeptide chain. Hemoglobin is an excellent example of quaternary structure of protein. See the pictorial view of the structure of protein, how the various amino acids are linked to each other to form the polypeptide chain, and that undergoes for the three-dimensional modification and forms the different types of proteins like secondary, tertiary or quaternary proteins. Proteins performs different functions in the body: chemical nature and solubility and nutritional importance of the protein. Functional protein, chemical nature and solubility and nutritional importance of the proteins. Functional classification of the protein. Structural proteins, example keratin, collagen. Keratin present in our nails, hairs. Collagen in the muscles. Enzymatic protein, pepsin. Transport proteins, hemoglobin. Hormonal proteins, insulin, growth hormone. Contractile protein, actin, myosin present in the muscles, helps in the movement. Storage proteins, albumin. Genetic proteins, nucleoproteins. Defense protein, immunoglobulins. Receptor proteins for hormones and viruses. Classification based on chemical nature and solubility. Simple protein, they are composed only of amino acid residues. Conjugated proteins, along with the amino acids, there is a non-protein prosthetic group. Derived proteins, they are denatured or degraded products of the above two. Proteins, simple, conjugated and derived. Simple example, globular and fibrous. Globular, albumin, globulin and histones. Fibers, collagen, elastin and keratin. Conjugated proteins, nucleoproteins, glycoproteins, mucoproteins, lipoproteins, phosphoproteins, chromoproteins, and metalloproteins. Derived proteins, primary and secondary, coagulated proteins, proteins, and metaproteins. Secondary, proteases, protons, and peptides. Enzymes are a group of catalyzing functions in a biological system. They are usually proteinaceous substances produced by the living cell without themselves getting affected. Enzymes enhance the rate of reaction and are formed in the cell under the instruction of genes. Enzymology is the branch of science that deals with the study of enzymes in all aspects like nomenclature, reaction, and the functions. Enzymes occur in colloidal state and are often produced in inactive form called proenzymes, zymogen, which are converted to their active forms by specific factors like pH or substrates, etc. The enzymes that are produced within a cell for metabolic activities are known as endoenzymes, and those which act away from the site of synthesis are called exoenzymes. So enzymes are nothing but that is the complex proteins and performs various functions in our body. General properties of enzymes and factors affecting their activity. Enzymes accelerate the reaction but do not initiate it. Enzymes themselves do not participate in the reaction and remain unchanged at the end of the reaction. Enzymes are therefore needed in small amounts. The molecules of an enzyme is larger than that of substrate molecules, and hence during reaction a specific part of enzyme molecules comes in contact with the substrate molecule. That part is called active sites of enzymes. Amphoteric nature of enzyme. Chemically, most of the enzymes are proteins and therefore show amphoteric nature. The enzymes can react with acidic substances as well as alkaline substances. Specificity. Most of the enzymes are specific in their action. A single enzyme acts upon a single substrate or a group of closely related substrates. For example, the enzymes urease can act only upon urea invertase and can act upon sucrose only. A slight change in the configuration of the substrate molecule requires action by a different enzyme. Colloidal nature. All enzymes are colloidal in nature and thus provide large surface area for reaction to take place. Colloids are a mixture of two components, that is dispersed particles and dispersion medium. The size of the dispersed particle is larger than dispersion medium. Enzyme optima. Enzymes genetically work best under certain narrowly defined conditions referred to as optima. This includes appropriate temperature and pH. Temperature sensitivity. Since the enzymes are proteins, they are affected by change of temperature. With the increase in temperature, increase in the enzyme activity takes place up to 40 degrees Celsius. However, when temperature increases above 60 degrees Celsius, the proteins undergoes denaturation or even complete breakdown. When the temperature is reduced to freezing point or below freezing point, the enzymes becomes inactivated, but they are not destroyed. The rate of reaction is more at optimum temperature. pH sensitivity. Most of the enzymes are specific to pH and remain active within particular range of pH. The strong acid or strong base denature enzymes. Most of the intracellular enzymes function best around neutral pH. Concentration of enzymes and substrates. The rate of reaction is proportionate to the concentration of the reacting molecules. If the substrate concentration is increased, the rate of enzyme action also increases up to the certain limit. Beyond a certain concentration, the enzyme's molecules remain saturated with the substrate molecule and the activity becomes steady. Enzymes inhibitors. Enzymes inhibitors are certain products which inhibits enzymes activity during the reaction. If the active sites of enzymes is occupied by these inhibitors instead of substrates, molecules and the activity of the enzyme is lost. These substances are like substrate molecules in their structure are called competitive inhibitors. See the role of enzymes, how the enzymes decreases the activation energy and that helps to accelerate the rate of reaction. Yes, in the optimum pH the rate of reaction is maximum, at optimum temperature also. In the third, if the substrate concentration is increases, the rate of reaction also increases, but after some time that increased substrate concentration having no effect on the rate of reaction.